Hello and welcome to Close to the Edge, the new series from Gen Edge, where we invite chief executives and outstanding scientists from groundbreaking biotech and pharma companies to sit down with us to discuss their science, their technology, and their business strategy. I'm Kevin Davis, editor-at-large with Gen and the author of Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing, out now in paperback.
And I'm Alex Philippidis, Senior Business Editor with Gen, Genetic Engineering and Biotechnology News, the publication covering the biotech industry for 40 years. Close to the Edge is an offshoot of Gen Edge, our new premium subscription channel from Gen, providing in-depth exclusive news, interviews, and analysis of key trends in the biotech industry, coupled with a range of multimedia offerings such as this one. More details of our free trial offer are at www.genengnews.com/genedge.
On today's episode 8 of Close to the Edge, we welcome Sujal Patel, the CEO of Nautilus Biotechnology, a 5-year-old Seattle biotech developing a large-scale single-molecule proteomic analysis technology capable of delivering, it says here, extreme sensitivity and scale. Nautilus has ambitious goals of democratizing access to the proteome and enabling fundamental advances across human health and medicine. Sujal, great to see you again. Welcome to Close to the Edge.
Thank you, Kevin and Alex, and thanks for having me here today.
You're welcome. First off, Sujal, how does Nautilus define democratizing access to the proteome?
Democratizing access to the proteome. So I guess first we have to sort of back up for a second and just level set on what is the proteome. And, to do this by using an analogy to a world that we understand, the genomic world has largely been conquered over the course of the last 2 decades. Today, we have democratized access to the genome. If I take a drop of your blood, Kevin, and I want to understand what DNA is in that drop of blood, I can get you 99% of the answer for less than $1,000, and it just takes a couple of days. And it's a push button instrument. Illumina is the leader in that space. You push a button and out comes the analysis. If I want to answer a slightly different question, what proteins are in that drop of blood? The answer is much more complicated. Today's best workflows for telling you what proteins are in that same drop of blood involve a complex set of sample preparation and fractionation and digestion steps. They use an instrument called the mass spectrometer to perform measurements, and there's a complex set of bioinformatics on the other side. And after doing all of that work and spending weeks and weeks and maybe even $10,000, the very best that we can do as humanity is maybe identify about 8%, single digit, 8% of the proteins in that sample. That's a really big problem. It's a big problem because over 90% of our FDA approved drugs target proteins. Most of our molecular diagnostics target proteins. And so that inability to measure proteins in sample has been a huge impediment to biopharma and the development of new and more effective therapeutics. It's hampered this precision and personalized medicine wave that we've talked about for many years. And for us, when we say democratizing the proteome, we mean producing a new instrument based on new science and new technology that takes sample in and returns virtually the entire proteome from that sample in a simple push button manner, just like we've seen occur for genomics over the course of the last 15, 20 years.
Fantastic. Well, we look forward to hearing much more about that technology and what you plan to do with it during the rest of the program. First, Sujal, let's sort of just wind the clock back a little bit. Some of our viewers may know you from the great success you had leading a company called Isilon, a large-scale data storage company. What did you do? Tell us a little bit about your background, how you launched Isilon, and a little bit about that story, which owes, I think, a fair bit of its success to, as you were just saying, the great progress we've seen in the genomics space over the last 10, 15 years.
That's right. Yeah, so let me kind of describe my background because it's atypical for the founder and CEO of a biotech company. Yeah. I'm a software engineer by training, and for the first 2 decades of my career, I was focused on the tech world, and in the last 5 decades, I've pretty much completely flipped over to the biotech world. The last company that you mentioned, Isilon, was a company that I was founder and CEO of. It was founded over 2 decades ago. And over 2 decades ago, the world was in a transition. It was in a transition in the enterprise from structured data like text and database data, transactions like your credit card transactions, to unstructured data, video, images, graphics, machine generated data. And that transition required a new storage architecture, and we built a new product which provided a new storage architecture for enterprises. And when we first went to market in 2003, we started with a pretty verticalized approach. We went after media companies and internet streaming companies that were putting audio and video on the internet, and areas where this problem was prevalent first. And the company grew very nicely from that point. By 2004 and 2005, one of the big things that had happened was that biomedical research and life sciences was driving a huge amount of our business because their storage needs were exploding. And they were exploding because of a number of reasons. Genomics was the primary driver. And if you recall, kind of 2007 is when Illumina bought Solexa and really kicked off this NGS wave that has led to an entire ecosystem in genomics today. That ecosystem really helped our business because it was data intensive, and it was exactly what our product was built for in the last company. In addition to that, proteomics, mass spec-based proteomics was a driver. In addition to that, high resolution microscopy and other sorts of technologies that were helping to move science from the laboratory into the data center. And so that was a big piece of our business for many years. In fact, some quarters, we took that company public, my last company, in 2006. And for many of our quarters as a public company, the biomedical research life sciences vertical was the largest vertical market that we were selling into. Yeah. And so it was an area that we got to know really well. Yeah. If I kind of continue describing my background, we ran that business till 2010, and I was CEO up until the end of 2010. Q4 is when we sold it for $2.6 billion. The company was quite successful by that point. We were just coming off, we never reported that last quarter, but it was almost $100 million quarter, and very profitable as well. The business had a 20% operating margin that quarter. The whole year was profitable for us. And that's gone on with the acquirer to become a multi-billion-dollar business. Yeah. I stepped out of that business in 2012 at the end of the year, and really tried to focus on what I wanted to do next. And for me, I have always had a great deal of interest in biomedical research, and a lot of that was spawned from what we saw at Isilon as I was dealing with these customers, and it became one of the 2 philanthropic focuses for myself and my wife as well. So I've gotten to know the space pretty well, and I started thinking about ways to get involved in that market or in clean energy. Really, I was looking for something that was very different and very impactful on people and society. And an interesting connection here for you is that when we started Nautilus in 2016, my co-founder is a guy named Parag Malik. Parag, I've known since those old days of my last company. He was my customer in 2004 when he was running clinical proteomics at Cedars-Sinai Medical Center, and I got to know him really well. About a decade ago, he went to Stanford to start a new lab sitting at the intersection of data science and computer science and biochemistry, and he has academic degrees in both. His lab sits at that intersection. My wife and I were so impressed with his work at Stanford. We've been philanthropically supporting his lab for a decade. So that built a close relationship between us. Yeah. And in 2016, he called me up and said, I figured out how to crack this proteomics problem, and I want to tell you about it. And in about an hour, I was at the edge of my chair, ready to just dive in. It didn't take us more than a week to hammer out how we were going to go do this together. And it has been an incredible ride over the course of the last 5 years.
That's fantastic. So how does you obviously had success with Isilon, as you've just very nicely summarized. How does that business and commercial success translate now into your new venture at Nautilus?
Yeah, so a lot of it translates, and a lot of it I've had to learn, right? Okay. So what translates? This is a business like any other business. It has a value proposition. We have to build a product and technology. It's got lots of different pieces. There's complexity. We have to scale a go-to-market model that actually is very, very similar to the go-to-market model that I used in my last company. There's direct sales force. There's all of the same jobs underneath it. Maybe the titles have changed here or there, but there's a lot of similarity there. And so, for me, a lot of the lessons that I've learned have translated directly into helping this company grow very quickly and get through its research phase quickly. Some of the areas that are different are just kind of what you would expect, right? I mean, I walked into this company and I had to figure out what all these terms meant in biotech. How does biology work? What about biochemistry? How do I figure out all the various pieces that we have to put together? And it was a long process of learning over the course of the first couple of years as I ramped up on the biology and the chemistry and in biophysics and the whole range of other disciplines side.
And proteins are so darn three-dimensional, aren't they? It's a little more complicated in some ways than just managing A's, C's, T's, and G's.
That's right, for sure. I mean, there's a lot more complexity in proteomics, which has made it difficult to measure proteins in sample, why this breakthrough has never occurred.
Yeah.
And then, in the genomics era, we borrowed from nature. We borrowed techniques to copy and read DNA from nature, which is necessary for cell division. And there are no techniques to borrow in proteins. Once something becomes protein in nature, it can't be read, it can't be copied. And so these are all new techniques that had to be built. And it took a while for, I think, the world to have the pieces together that enable this type of revolutionary approach.
Right. Alex, over to you.
Thanks, Kevin. I guess, how did the company you created come to be called Nautilus?
Well, that's an interesting question. How did the company become Nautilus? So we started with, like most startups do, a working name. Ignite Biosciences was our working name, but we had to come up with a name that really spoke to us and was connected to our mission. Many of you probably recognize Nautilus as the name of that many submarines carry, and it was actually one of the first submarines. And it was the submarine in Jules Verne's novels as well. And so, first and foremost, we had this submarine that's something that comes out of that word Nautilus. And a submarine is about going and diving deep and getting access to things that you don't see if you're sitting on the surface. And so, going and digging deeper into the proteome was a really interesting analogy that we thought was relevant for our mission. The second thing is that the Nautilus shell, in terms of the sea creature, the Nautilus shell is a logarithmic spiral. And if you look at it, the logarithmic spiral is very pronounced. And the core of the approach that we use to quantify proteins in sample is rooted in data science and statistics and machine learning. And we really felt that the parallels between this mathematical representation in the shell and the core of our technology was really a great parallel as well. And so for those two reasons, we named the company Nautilus, and you sort of see in our logo that the periscope and that sort of evocative shape that we have kind of signifying the root of the name.
And speaking of proteomics, which you've begun to do, what's your take on the state of the field? For years, proteomics has been kind of the Rodney Dangerfield of genomic sciences, seeming to get no respect. How much has that changed in the years you've run Nautilus, and why do you think that is?
Well, so I think that I might structure the question a little differently. I think genomics has been so big of a part of the conversation in the scientist community because we figured out how to measure it easily. We figured out how to measure it inexpensively, and there was a lot to be learned from it. And so, when genomics really came into the limelight, I'll call that kind of 06, 07, 08, it was really unknown how we were going to leverage the genome to go and impact positively human health. And so a lot of those early sales of NGS instruments started in academic and research. People were trying to gather the data because they could, and proteomics was still really, really hard. And so very, very few labs in the world could do a good job at proteomics. And so everyone gravitated towards genomics. Fast forward to where we are today, genomics has become a really big thing. And there's a whole ecosystem of companies that are surrounding the NGS instrument vendors. What the scientific community realizes today is that genomics is great, but it's not enough. We need to be able to understand what's going on at the protein level. So we understand what is going on inside of a cell. The proteins make up your cellular machinery. We have to understand that. And there's a lot of scientific data out there showing that there's poor correlation between gene expression and what actually is going on at the protein level. And so as the world has gone through that recognition, and as biopharma companies have begun to push the bounds of current mass spectrometry-based proteomics and current assay-based proteomics, there's certainly, at least from our standpoint, from talking to a lot of these potential customers, there's certainly a real desire to go and have a next generation instrumentation platform that can do for proteins what Illumina largely did for the genome. And that's really what our focus is. And we think that when you can get to that point, it really helps those customers in biopharma develop better diagnostics, accelerate and reduce the cost of therapeutic development, make more effective drugs that have less cross reactivity. We feel like there's a lot of transformational use cases that are right in front of us. And as the proteomics field has evolved and new companies are promising new approaches, there's a lot of interest in tracking them and trying to figure out, which one of those companies will be the winner next.
Well, let's look at Nautilus as it's sized up in simple terms. The proteomics platform of your company is a large-scale single molecule platform, potential to achieve quantification of more than 95% of the proteome. What does that mean and how does that work?
So let's talk about what it means first. So if you take a sample, let's use 100 to 1,000 cells. I use that not by accident. 100 to 1,000 cells is a common sample type that's used across the entire range of therapeutic development workflows. 100 to 1,000 cells has somewhere on the order of 10 billion protein molecules in it. And if you want to go and effectively understand what's going on in those cells, you need to understand and identify every one of those proteins. You need to figure out how are they modified, what form are they in, because all of those little differences affect the protein's function and affect the cell's function. And so, we set out 5 years ago with this idea of, okay, let's get to the point where we could identify all of the proteins in a sample. So instead of being able to identify only 8% or 1/3 of the proteins that make up a human, be able to identify almost all of them. And so we set out in 2016 and 2017, and we went out and had many conversations with biopharma organizations, and that ramped up. We had 50 by the time we got to the end of 2017. We've had over 150 now. And those early conversations informed us what was the criteria that biopharma and diagnostics companies needed to really do the type of research that they wanted to do in the protein space. And we heard a lot of interesting things. We heard that the instrument needs to be incredibly sensitive, which led us to the requirement that this needed to be a single molecule platform and a single protein molecule, not a peptide molecule. And we may loop back to that later, what that differentiation is and why that's important. We also realized that the product needed to have nearly complete coverage of the proteome because any hole means that you're missing a piece of the data, and that could be the critical piece of the data. And because proteins operate in networks, if you missing a little piece of data, the next downstream effect might be missed, and then the next downstream effect might be missed. And so these gaps will amplify over time. We realized that our customers truly needed an easy to use solution. Today, proteomics is trapped in a handful of the very best labs in the world when you talk about trying to identify the most proteins from a sample. It's kind of discovery proteomics world. Many customers need a push button solution that works in an easy way so they can just do good science, not have to become experts in a mass spectrometry based proteomics workflow. We also realized from talking to customers that dynamic range was really important. And this was a key design criteria for us. Alex, you described it as a massively parallel system, I think. That really was something that we took to heart very, very early. 100 to 1,000 cells has 10 billion protein molecules. Our customers want to be able to identify all of them. And so even if somebody else makes a solution that identifies, 80% of the proteins that are in the human proteome, can they analyze 10 billion molecules at the same time in a single run of the instrument? For many of the customers that we talked about, they wanted the flexibility to reach that dynamic range. And that was one of the key stretch goals for the platform that took a lot of engineering and a lot of science to go figure out how we were going to go do. And so, when we talk about sort of, what we've designed the platform for, those are the big criteria that we've designed the platform for. And really, if you look at what we're talking about bringing to the market here over the course of the next couple of years, it is basically that list of design criteria that customers told us about a couple of years ago, or almost 4 or 5 years ago, starting then.
Nautilus has said its platform could unlock some high value applications in precision medicine, drug discovery, and diagnostics. Beyond simply identifying proteins, can you share some examples of some applications that this would enable?
So let's use some concrete examples, both on the diagnostic side and the therapeutic development side. If you think about therapeutic development, the first step is target discovery. Let me go take 1,000 cells that are healthy, take 1,000 cells that are afflicted with some disease. Let me go analyze the proteins in both of them. Let me find what the differences are, because those differences could be targets for drugs. They could be things that we want to modulate in the cell to be able to achieve a positive impact on the patient who's afflicted with this disease. This process of target discovery today is really hard because the traditional proteomics approaches only return a small fraction of the proteins that are there. And some of the approaches in the proteomics world return the most abundant proteins that are there. The problem is that the things that differentiate healthy and sick cells, they're often the least abundant things that are on a cell surface, for example. And so first and foremost, we look at our technology when we talk to our potential customers in biopharma, we look at our technology slotting into that first part of the therapeutic development workflow and finding better biomarkers for our customers to go after. From there, there's more questions the customer wants to answer. So now I've developed a library of potential compounds that I can use to influence the behavior of a disease cell. Well, how does that work when I introduce that compound to a cell line? What happens? What are the downstream effects? These mechanism of action studies to understand what's going on are really, really difficult to do today because we have such poor coverage of the proteome out of the samples that are used in that workflow. Follow that train down the next one. Okay, I've got a compound that I like. Well, how toxic is it to the body? What happens if that compound interacts with a cell line from the liver and from the kidney and from your heart? Where's the cross-reactivity that's going to go and take this drug and cause it to fail in the last stage of clinical trial before it goes and hits market after I've invested billions and billions of dollars? Let's back up in the process. Let's take all those cell lines from the human body. Let's expose them to these compounds. Let's see at the protein level what changes have occurred. So, as we start talking to customers, we really see like the light bulb turn on as they try to think, okay, I've only done it this way, but if I had complete proteomic information, if I could get that routinely with push button instrument, yes, it would be transformative to a lot of those different workflows. Same sort of thing on the diagnostic side. If you want to figure out if a therapeutic is working or not, if you want to figure out, should I give somebody therapy A or therapy B, the standard is to go look at protein biomarkers in blood, in cells, in any type of biofluid. And today's technologies are just not sensitive enough to dig in deep and figure out where are the biomarkers that are truly going to be indicative of therapeutic response, that are truly going to tell you which of the potential drugs that are my options should I choose. And so it's a very similar use case over there that's enabled by this wide dynamic range and sensitivity of our platform.
Does Nautilus intend to evolve into either a drug discovery company or a diagnostic company down the road, or would you just be happy to partner with other companies in those fields?
Yeah, so this is it's a big question that we asked ourselves very early in our company's life. And I think that, the guiding principle for us as a company is that we want to build an instrument that we can get into every lab in the world so that any scientist who wants a proteome can get a proteome with the push of a button. And we feel that not only does that help have the biggest impact on human health because we make the instrument ubiquitous and the technology ubiquitous, but it also creates, in the long run, an incredibly valuable business and a huge business opportunity. And so that is our primary focus. In terms of therapeutics and diagnostics, who knows what we will like to do in the long run, but certainly in the short and the medium term, we're really only focused on partnership opportunities there and really trying to work with our potential customers to advance their science to whatever degree they want to work. Some customers want the instrument and they want us to give it to them and give us a service that helps them manage their data and analyze their data, and that's good. Some want to partner deeper. They have more custom questions. They want some help in terms of data analysis and machine learning. And so we're trying to figure out exactly how to meet our customers where they want to be.
Interesting. Over to you, Kevin.
Thanks, Alex. Sujal, Nautilus, my understanding is your goal is to commercialize your platform. It's probably in the next 2 years, I think late 2023. What is that correct? And what are sort of the key milestones that you need to hit in order to meet that timeline?
Yeah, so happy to walk through that. So I think that many of you probably know that Nautilus went public earlier this year, and as part of that process of becoming a public company, we have outlined a set of milestones for our investors to track our progress against over the course of the next 2 years. Today we're still really a pre-commercial company. We are doing we have started doing some analysis of basic samples with our first collaborator, Genentech, and we'll continue to work with them as well as add other biopharma organizations here over the course of time. But the big milestones are really in 22 and 23. Next year in 2022, we intend to pass through the milestones of being able to identify about 2,000 proteins per sample in the first half of the year and get to about 10,000 proteins per sample by the end of the year. And just for reference, there's roughly 20,000 base proteins that are encoded from genes in the human body. So that's about half by the end of the year. And then by the middle of 2023, we will get to about 95% coverage, which we consider to be comprehensive. The model for our customers to engage with us during the timeframe from today to the middle of 2023 is really a model where the customers send us samples, we analyze them for them based on some sort of partnership or collaboration, we give them results, and we really work very closely with them, understanding the data coming off our platform in the very early days of our platform's evolution. As we get to the end of 2023, that's really the point where we launch an instrument. And the instrument marks kind of a little bit of a shift in our business. Largely from that point forward, our revenue streams are selling instruments, selling consumables, selling software. And really for us, there'll be some modest revenue that comes off of some of the deals that we do earlier, but that's the big catalyst for the revenue ramp for this company, is being able to get that instrument out into the world and making it push button simple for any scientist who wants a proteome to get a proteome.
Right. You mentioned, Sujal, taking the company public a few months ago through a SPAC deal. Was that always the plan, or did you take advantage of, did a window open that you decided now was just simply too good an opportunity not to take advantage of?
Yeah, I mean, it's a good question. Having been part of, my second startup that I've been running, third startup that I've been a part of directly, and like 70 or 80 that I've invested. And I can tell you that the headlights don't reach very far in startups. They have plans that go out a year or two, and then there's lots of things that change. For us, we transitioned at the beginning of this year out of the phase of research into the phase of formal development and manufacturing. Our costs were going to go up quite a bit. And even though we hadn't started spending our Series B funding, which we raised in early 2020, we were being approached by a lot of investors to go and put more money into the company. And at some point, I stopped letting the calls go to voicemail and I answered the phone. And I realized that there probably was a deal to be had in the private markets that would be interesting for the company. And as I started talking about that with my board, another opportunity came up. One of my board members is a man who is named Mike Altman, and Mike's with Perceptive Advisors, which is one of the very, very best life sciences hedge funds in the country. And they're a prolific SPAC sponsor. They're onto their 5th SPAC right now, and they've been very successful in that world. And Mike brought an opportunity to us that really fit our goals, right? It gave us even more capital to accelerate and de-risk our timelines. It got us out as a public company, and it did so with a partner that we knew really well. And Perceptive was an investor in the company already. They'd been on our board. We were very comfortable with them as a partner, and so it was a great opportunity that we took advantage of, and we were able to get out in a very nice timeline. And it left us with a balance sheet that was really significant in terms of our ability to execute on our development and commercialization plans. We ended last quarter with $388 million of cash on the balance sheet, which is a great place to be.
That's great. What surprises or challenges or hurdles have you encountered since taking the company public? You've got to keep your investors pretty happy, right? That's quite a. You said the headlights don't go very far, but you're hoping they go at least 2 years to hit that magic commercialization juncture that you mentioned.
Yeah, for me, this is this second time around as a public company CEO. I don't think there are really any surprises or challenges here. For us, the important things right now are to go and take the larger investors in our company who are long-term healthcare stockholders, educate them and continue to educate them on the evolution of the proteomics world, make sure that they're up to date on the progress that we are making as a company, and highlight for them the things that they should look for to indicate that we're on track, and start building a track record with these investors, right? We highlight the fact that they should look for a set of publications that we've described that show our progress. We told them to look for our partnerships and collaborations, not just, that how many we have, but who they are and are they important use cases, and then really watch our progress towards those major milestones that we talked about. And so for us, those conversations are going well. We don't control markets. Markets are very wrong in the short term and very right in the long term. And so we're really long-term focused here and just trying to build those early relationships as we've now fully transitioned to a public company.
Yeah, fantastic. Alex, over to you.
Oh, thanks, Kevin. Now, speaking of investment, one of Nautilus's investors has been Jeff Bezos through his Bezos expeditions and later via Amazon. We know Bezos has reportedly invested in an anti-aging startup called Altos. Is Jeff personally interested in proteomics, and what have your dealings with him been like?
So I think I will just talk briefly about sort of what I know about his interests and leave out the details of it. But, Jeff personally has an interest in healthcare, in bettering human health. And you've seen when he was CEO of Amazon try a lot of different things related to health. Jeff has, if you looked up the Bezos expeditions investments, he's made investments in numerous life sciences companies. And certainly, being based between Seattle and Silicon Valley, it's a small community here, and certainly we were excited to have him as an investor in the company.
Now, Jeff Bezos is one of several name investors in Seattle. He seemed to have captured the market there because other institutional investors Nautilus has had is Vulcan Capital with Paul Allen and Madrona Venture Group. I know proximity to major investors is one key advantage to being based in Seattle. What are some other advantages that you found?
Well, first and foremost, I would highlight the reasons why I'm in Seattle, right? In 1996, I had spent my whole life on the East Coast. I grew up in New Jersey. I went to school in Maryland, and I wanted to come to the West Coast. I wanted to come to the West Coast because that was the mecca of technology. And I interviewed in Silicon Valley and I interviewed in Seattle. And I liked Silicon Valley, but it was a big valley, strip malls, and some tech companies peppered in. It was okay. I came up to Seattle, and it was a vibrant city. It has a beautiful bay and multiple lakes. There's a lake right outside my window here right now. There's so much access to the mountains and the outdoors. I was really captivated by the ability to pursue a career in tech and then also enjoy this city. And what's happened over the course of the 25 years since I came here is a massive tech industry has been built with multiple mega cap companies, many mid cap companies, lots of startups. There's a vibrant community in tech. And now that same thing is occurring in biotech. We have, companies like Seagen and Juno Therapeutics, which sold for nearly $10 billion, and Adaptive Biotechnologies, NanoString, and many other companies across the range of therapeutics and DXN tools. And there's this vibrant community that's being built that I think over the course of the next 10 to 20 years is in biotech will be just as big as tech is. So for me, this is just a great place to live, and it's a great place to do business.
How closely do the two communities, tech and biotech, intermesh? Do you see whether it's partnerships or activity between the two, or are they really more in silos?
So we are in a phase of evolution in biotech where more and more data is being generated, and we're looking for better ways to analyze and make sense of that data. And so with that, the intersections of computing and data science and biochemistry, those intersections are getting tighter and tighter. And with that, tech companies and biotech companies are collaborating more computer scientists are starting biotechs, and some of the investors like Madrona Venture Group, which you mentioned a minute ago, some of those investors have begun to diversify from just tech portfolios to also biotech. And so if you use Madrona as an example, we were not the first biotech investment that Madrona made, as even though the vast majority of their portfolio is tech. And after us, they've invested in a number of biotech companies. They've invested in TwinStrand Bio and A-Alpha Bio, both based in Seattle here, and a number of others. And so we think that that's going to continue. And we think that given that, from my vantage point, that biotechnology and healthcare in general is really the next big frontier, I think we're going to continue to see that trend over the course of the next decade.
Another key resource in Seattle is people. So I want to get your sense, Sujal, what is the company's current headcount, and how much is that likely to grow over the coming year or so?
Yeah, so I mean, the headcount of the company is roughly 100. It's a tight group, really focused scientists and engineers who are trying to get this technology out into the world. There's a lot of growth in front of us, and just as an aside, about a quarter of our headcount's up in Seattle, and about 3/4 of it is in Silicon Valley, and that'll change a little bit over time, but that's what the mix is right now. If you kind of look at the hiring that's in front of us, over the next 6 quarters, we have something on the lines of 150 adds to the company as we really begin to not just push through the development phase, but really start to build the commercial muscle in this company as well.
I know Nautilus has strengthened its executive muscle this year. It's hired Carl Voss as VP of Life Science R&D, Matt Murphy as General Counsel. Where do you see the need to strengthen the company going forward? More on the senior level, or will it go more toward R&D perhaps?
I think that, out of the large growth that's in front of us, we've got growth at all levels. Individual contributors, exceptional scientists, research associates, middle-level managers, senior managers. And so there's a lot of growth to happen across the company. And there's always a big transformation in companies like ours as you move from pre-commercial to post-commercial phase. I will say from an exec team perspective, I'm really pleased with the fact that over the course of the last, really 18 months in earnest, we've built an extremely seasoned executive team, and one that's functioning really well. And the team draws from 2 roots. Some part of the team, outside of the 2 founders, some part of the team worked with me in the past at my last company. So Anna Maury, our CFO, Mary Godwin, our head of operations, Chris Blessington, our head of marketing, all came out of my past at Isilon. And then other folks like the ones you mentioned, as well as Nick Nelson, who's our chief business officer, and Super Sankar is our SVP of product development. Those folks all came out of the NGS revolution. And so we think that the marriage of bio talent and tech talent, the marriage of this NGS talent and the experiences that I've had in the past really create a very unique team that's well positioned to execute in the next 4 or 5 years.
Interesting insights on people, and more coming up. Over to you, Kevin.
Thanks, Alex. Sujal, Alex mentioned Carl Voss, who's obviously a pretty important cog in this machine. You recruited him from PacBio, whose CEO, Christian Henry, incidentally, was our first guest on Close to the Edge a few months ago. So Carl is leading the team to deliver Nautilus's first generation product. How do you see his role, and how is the team coming together to deliver that goal?
Yeah, and so I'm happy to kind of talk through that. I mean, Carl is a really incredible individual. In his 13 years at PacBio, he ran at some point or another pretty much the entire R&D organization. And he saw that company evolve from a phase that's probably a little earlier than where we are today, all the way through commercialization and scale. And he's got a tremendous skill set, PhD in chemistry, and just a tremendous skill set and experience from having been through that journey. He's paired with Subra Sankar, who's our SVP of product development. Subra was the guy who joined as head of engineering at Solexa prior to the acquisition by Illumina. And the acquisition of Solexa was what got Illumina into NGS in the first place. And after it came onto the other side and was at Illumina, he shipped many of the platforms afterwards, MySeq, the CBOT, CBOT2. And so the combination of those two folks really bring together all of the disciplines that we need to get this first product out. There's chemistry, there's probe development, there's all of the different various pieces of biochemistry that we have that feed into the assay. There's the assay itself, there's the instrument, and then of course there's the software and bioinformatics that marries up with that. And so, those two guys are two of the key leaders that are responsible for the delivery of that whole system.
Yeah. We also noted that you also recruited a scientist from Sweden named Emma Lundberg to the scientific advisory board. She's a professor in cell biology, proteomics. What made someone like that, one of the real world leaders in proteomics, so excited to partner with you and help you along the way?
Yeah, I mean, and we're really fortunate to have Emma on our SAB. Emma joins already a group that we're really fortunate to have affiliated with the company.
Yeah.
Lee Hartwell, who's a he's a Nobel laureate and
and always
will
will
be.
be.
The proteomics world. Rudy Aebersold is well known as kind of as the father of the proteomics world. Josh Lebert is the head of the Biodesign Institute at ASU and was the former president of the Human Proteome Organization. And so we're really excited about having an SAB who is really known as the key opinion leaders in the proteomics world. Why are they here? I think they're here for the same reasons that I'm here. First and foremost, we're here because my co-founder Parag is a KOL in the proteomics space. And he is widely regarded as a guy who thinks about problems in a creative way and does good science. And had anyone ever brought me a proteomics idea to go invest in, I would have shown it to Parag and said, Parag, what do you think? And here it is, Parag's bringing me the idea.
Right.
And the second thing is that when we describe the concept to our SEB members to try to recruit them, we describe them to customers or partners. They all, once the aha moment comes when they realize how this approach works, they immediately realize, okay, this is different than what else has been tried in the past. It's different than what other companies are doing. And I see how this can help to get to a solution that truly will help democratize proteomics. And once that light bulb goes off, it's usually not such a difficult task to get them across the line.
Yeah. Closing couple of minutes here, Sujal, but interesting that you bring up, the sort of what differentiates you from other companies. You're certainly by no means the only company that has expressed interest in quantifying or identifying the human proteome. I think at Gen Edge, we've had at least a couple of stories in the last 12 months of companies that dare to claim that they want to be the Illumina of proteomics or whatever it is. So do you see this space becoming even more competitive over the next 2 or 3 years? Or are you still really confident that your technology is the one to beat?
Well, I see the world becoming more competitive, but let me describe competitive, right? In any market where there's a great deal of interest and there's a desperate need for innovation, there's going to be lots of companies. If they're not, you should be looking at yourself and wondering, well, why isn't there a lot of competition? And there are hundreds and hundreds and hundreds of millions of dollars. Maybe, I added up, it's over $1 billion, I'm sure. Actually, I can add up 2 companies that get over $1 billion. It's well over $1 billion of funding that's recently gone into new proteomics companies that are trying to make a dent in this world. And so we think that there is a lot of innovation going on. There are going to be competitors out there. But what gives us a lot of comfort is that, going back to what I described earlier, we set out 5 years ago with the customer in mind and really trying to understand what was going to make a dent in therapeutic development, what would really be transformative for precision medicine. And we've been laser focused since this company was founded on building to those specifications. And we have a very unique approach. Most of the newer companies that talk about protein sequencing are focused on using variants of Edmond degradation, which is an old scheme to sequence peptides, not full proteins, and trying to make that more parallel. It's a hard task, and it's one that would not have been able to reach all of the design goals that we set out for. And I think, we describe it on our website and our material, but our approach is different. Our approach takes a molecule and probes it repetitively with getting very fuzzy data points and combining those computationally to a shockingly specific identification that this is an EGFR molecule, this is MEK1, this is GRAC6. And that is a very unique concept, but one that is very common in computer science. And it's really this combination of computer and data science and biochemistry that came together to come up with the core innovation behind this company. And that's a pretty rare thing, but something that Parag, my co-founder, he's got academic degrees in both. His lab at Stanford is in both worlds. He's been doing that his whole career. And so he was uniquely suited to come up with that idea. And the world has uniquely evolved to give us sufficient computing power in the cloud to do what we're describing for billions of molecules in a run.
Yeah. Ironic that you're in the cloud now.
Yes, again.
You said a few minutes ago, Sujal, in closing, that your company will be growing quite aggressively over the next couple of years and hopefully beyond. What sort of folks are you looking for in particular? Do you have any specific needs on either the wet lab or the IT front or the engineering front?
We need everything.
Okay.
I need organic chemists and single molecule biophysics, physicists and nanofabrication engineers and software engineers. I need to have accountants and recruiters, marketers, salespeople. I think the core thing that we look for is we look for people who are driven by our mission, right? We as a company have a mission to revolutionize biomedicine, and people who want to join us on that journey have the passion to go and do that. Those are the types of people that are really successful in this company, and people who want to drive hard and move fast, right? We're trying to do something really impactful and really hard. And there are lots of people in the world that are attracted to that. And if you've got those core tenets, at some point we'd like to have you work here.
Well, if I ever write a follow-up to cracking the genome, i.e. cracking the proteome, I hope that you and Nautilus feature in it. Really good chat, Sujal. Alex, I'll let you close things out.
Sure. Thanks, Kevin. And that's all the time we have for this episode of Close to the Edge. Thanks to our guest this past hour, Sujal Patel. We wish you and Nautilus all the best going forward. Check out Gen Edge for other Close to the Edge interviews with outstanding chief executives, including John Evans of Beam Therapeutics, Daphne Zohar of PureTech Health, serial entrepreneur Nessun Birmingham, and Ted Love of Global Blood Therapeutics. Gen Edge is where biotech gets down to business with exclusive in-depth reports from the Gen team. Please consider a free trial subscription, and you can find Gen Edge on Gen's website, genangnews.com.
Close to the Edge is produced by Hannah Turner and Liana Jabs. For Alex Philippidis, I'm Kevin Davis. Thanks for watching. Goodbye for now.